Hello? Yeah? Yes? No, I'll turn on my video. You can see me now. Yeah? Yeah, I'm connected. Don't worry. I do this every day. Yeah, I'm able to see everything. Should I start? No, I don't hear anything. Sure.
Good afternoon, sir.
Can you hear me?
Madam, sir, Madam is going to join. Lakshmi Madam is going to join within few minutes.
Okay. She just called me. She was not sure about this one, but I can hear you.
Yeah, okay. Just now we can see your. Okay, sir. Good afternoon, students. Last announcement. Please inform your friends. Let them join as early as possible. The section will be start within seconds. YouTube Live, Facebook Live, and Air meetings also coming. You can join anywhere for the section, but don't miss the section. So we'll wait for 2 seconds.
Sure.
లక్ష్మి మేడం మీరు స్పీకర్ గా సారీ పార్టిసిపెంట్ గా జాయిన్ అయ్యారు ఒకసారి లాగౌట్ అయ్యి మళ్ళీ జాయిన్ అవ్వండి మేడం ప్లీజ్ Yeah, madam. You had joined. Good afternoon, students. Today our speaker is Satyanarayan sir. His qualification is PhD. Since he is working from 2001 in Reddy's Lab, Hyderabad headquarters. Sir, heartily welcome to the section. You can introduce yourself.
Good afternoon. I hope you guys can hear me. Madam, can I get an idea of how many students are there so that I have at least one idea how many I'm speaking to?
Up to now, speakers are, students are 500, sir. They will
Okay,
improve the attendance.
cool, excellent.
YouTube and
Okay, sure.
Facebook also going on. They will come to there also.
Yes,
Overall, 600 students are there up to now.
excellent. Can I get started now?
Yeah, you can, sir.
All right, let me put the presentation on. I hope you guys can see my presentation.
Yes,
Uh,
sir.
all right, let me get started. So I was asked to present a little bit about biotechnology and vaccines. So what I will do today is I will cover the aspects of biotechnology from an industry perspective. Biotechnology is a very, very large field. So 2 hours or 90 minutes is never going to do justice to that. So I had to pick up specific areas that I can speak about with a little bit of confidence and give you an idea of how the biotech industry is panning out in India, what we do, how are we different from academics, so that you guys have certain idea about what the biotech industry is in India, what is your prospects, what are the things that are happening in India, and some part of it is pretty much the same outside India also. Okay, so let me get started. I've given the title from a cell to many billions is because one of the critical aspects of biotech is the fact that we usually work with cells. We manipulate cells, we take a single cell and then grow them up, do whatever manipulations we want to do with that, and therefore we produce a product from the cell. Now, the cell itself could be the product, but it doesn't matter how you look at it. Okay. So, what are the topics we should be considering today? One is I will be defining the problem that is there today from my perspective of what is the problem that we will be tackling. Then I will say how does industry solve these better than academics. And the reason I bring this up is a lot of you, I assume every one of you is a student in the biotech space, and you have learned biotechnology from an academic view. Now, is that view wrong? Absolutely not. But is that view sufficient? I don't think so. So academics focuses on certain aspects, industry focuses on certain other aspects. There is an overlap. There is no doubt that there is an overlap. But industry expects certain other things. And what I would like to do at the end of this 1.5 hour session is, if you can gain some aspects of how industry thinks about science, how industry does science, then I would have been successful in this lecture. Okay. The
Excuse
third
me, sir.
part of it will be, yes.
Please hide the button. Sharing.
Oh yeah. The third part is the challenges ahead and the roles for the next generation. I'm just going to give you a little brief about what you guys will be specifically seeing as areas that I think are of concern that you might want to think about, and what is your role as part of the next generation of scientists. And I will give a very brief view of the long term view of the field, how I think biotechnology, as students of biotechnology, how you should prepare yourself for the field. Okay, that's my thing. So let's get on with the presentation. What's the problem? What is the actual problem that we see? Okay, now I will start with a very simple question. What is the structure of a protein? If you go to the internet and take a look at the protein, if you go on a textbook, you look at the protein, normally you see something like this. And I give a very small image of a particular protein called GCSF, which is granulocyte colony stimulating factor. And I put up an X-ray structure of that one. Why did I put an X-ray structure? Because that's just considered the gold standard of structures of protein. But if you take a look at the structure, it has a certain number of, different. Uh, Let me use my laser pointer. 1 second. Yeah. So you will see that it has. This is the GCSF-R protein. This is a dimer. It has got a certain number of structures, and you assume that this is the final structure. But when I do the same search and say, don't give me an X-ray structure, but give me an NMR structure, this is the structure that comes up. Okay. Now, if you're paying attention to the structure, you will notice that there is not one structure but many structures in this. Actually, this one is a mixture of 10 different structures. Now, GCSF theoretically is supposed to have one structure that functions, but when you do the NMR, you get 10 structures. Are any of these 10 structures the actual structure that functions for the protein? We have no idea. Actually, nobody knows which exact structure is the functional structure. The reason this is the case is because proteins, unlike very small molecules, are not rigid molecules. They are very, very flexible. GCSF is especially very flexible. So you can understand that when somebody is trying to make a protein, they are only encoding the DNA. The DNA in a cell is then expressed. You end up with a protein and it folds naturally. Or you may have to artificially fold it. Once you fold it, is this the right structure? We have no idea. We assume that is the right structure, and that when we do a test in the lab to see if it is functional, as long as it's functional, we assume that we have achieved our goal. So in real life, when you try to say that I am trying to make a particular protein, you are trying to make a protein to the best of your capability. Why is this the problem? If you take a look at small molecules, the word we use very often is a generic molecule. In the case of proteins, we don't have something called generics. We have something called biosimilars. The reason it's never called a generic molecule is, if this is aspirin as a normal molecule, any company making aspirin will have exactly the same structure. Okay? Aspirin has 21 atoms. All the carbons, all the hydrogens, everything mixed is 21. Now you take a look at somatotropin, which is a protein, a small sized protein. It has 3,000 atoms, and you take any generic antibody, it has about 25,000 atoms, carbons, hydrogens, nitrogens, everything mixed together, 25,000. This structure, you can predict exactly which atom is going to be where in three-dimensional space. With this, it becomes very difficult. And just imagine when you reached an antibody, it's almost the same. The analogy given here is, this is a boat, this is a ocean liner. The size difference is huge. So predicting the structure becomes very difficult. Okay, is this the case only with protein-based things? Not really. Are vaccines any different? Okay, now everybody is talking about the infamous SARS-CoV-2 virus, and we're all interested in getting a vaccine. This particular protein which is sticking out is the spike protein, and here is your spike protein as an example. you will see that this is no smaller than an antibody, right? It's actually almost as big as an antibody. It may not be the same size, but it's close enough, in the same ballpark, which means the structure is not very well defined. That is the reason when you take a look at the kind of vaccines that are being created today, some people are making a viral based vaccine, some are making nucleic acids, some are just using a viral vector like adenoviral vector, and some people are using a pure protein. Each one is trying to find out, is my vaccine efficacious? You keep hearing it is 95% efficacious, right? Why are they saying it's 95? If it's a protein and you're injecting it into a animal, or in this case if you're taking it and injecting it into a human, you should be producing antibodies against the virus protein, right? But we don't produce the same kind of proteins necessarily, antibodies, and the protein you're injecting may not be exactly the identical one that is present on SARS-CoV-2. Therefore, we are always concerned whether we have a product which is of a nature that will allow us to give the right immune response. Okay, is this problem only associated with these vaccines and people who are making biosimilar molecules? The answer is no, actually. So, any biotech product, whether it is made by the innovator company or whether it's made by a biosimilars company like Dr.Reddy's, the product actually is, if you take the actual product in the vial that is sold to the patient, has a lot of structures. We try to find out what the exact structure is, but we are never capable of telling exactly how many structures exist in solution. So what this does is it always asks you the question, are all products biosimilars? Even the product is biosimilar to itself. It's a biosimilar to itself. And the reason, the distinction between a generic molecule and a biosimilar molecule is that in a generic, the structure is an absolute structure. Whereas in a biosimilar molecule, the structure is very, very, very similar across different batches, different lots. But nobody is the exact structure. The term, that's why it's called biosimilar. And if you buy an innovator's drug, one batch to another batch is theoretically also biosimilar. But since they are the innovators, that's called an innovator molecule. And since companies, in a lot of biotech companies in India make a generic copy of that, we are called biosimilars. Okay, that is in one sense the basic concept of biotechnology. We are trying to produce a product whose structure is not as well defined as we would like it to be, but we know that it works. So when I'm telling you this differences in protein, where are the differences coming from? What are the kinds of differences? And I'll just give you a little overview of the kinds of differences we are seeing. If you take this is an antibody structure. This is the what I would call the cartoon structure of an antibody, right? In reality, for those of you who are not aware of it, antibodies don't have this structure. When they are floating in your bloodstream, they are not floating as a Y molecule. They are actually folded onto themselves. This Y is mainly as a teaching tool so that students understand that there are two heavy chains and two light chains, and this structure is approximately in this manner. The linear structure is correct, that there is a long heavy chain, two long heavy chains connected by disulfide, and the light chains are connected to the heavy chain. All of that is true, but in solution, this is not the exact structure. Okay. What is the kind of stuff that is happening? A lot of antibodies on their N terminus have something called glutamine. In solution, when the N terminus is glutamine, it can become pyroglutamate, which is actually a cyclization of the glutamine. The amine group becomes glutamate, which is a cyclic glutamic acid. Therefore, its structure is slightly different. An amine group, if you remember your chemistry, is positively charged. Glutamic acid is an acid which is negatively charged. Suddenly you have shifted the charge of the N terminus from a positive to a negative. But because it's cyclic glutamic acid, it's called pyroglutamate. Okay. Then you have, if I were to go down the side, you know that asparagine has an second amine group, right? There is the amine that is there as part of the primary CON structure, the NH2 of the amine group of every amino acid. Other than that, asparagine amine can be lost. If you lose an amine, you become aspartic acid. Same thing. A positive charge of amine becomes negative charge. Suddenly you have shifted the charge of the protein. Isomerization can happen. An aspartic acid, if you know aspartic acid, has an carboxylic group. If you know the structure of an amino acid, a generic amino acid, you will say R for the functional group, COOH, NH2, right? So you got one amine group and carboxyl group. That second carboxyl group is normally attached to the main backbone, the peptide backbone. In aspartic acid, the carboxylic group can sometimes switch and become part of the peptide bond. If it does, it's now not aspartic acid, it's isoaspartic. This changes the structure of the protein completely. It's actually a very disruptive change. Racemization can happen. You know, all amino acids in human bodies are only L amino acid. If you try to eat a D amino acid, it will not be metabolized. Okay, I'm not very sure if it's poisonous, but it certainly is not metabolized. The only place in nature where you find D amino acids, rarely that you find, is on the cell walls or the cell membranes of bacteria. You heard of the beta lactamase, it breaks the DLR bond, the alanine, the D-alanine, that's a D amino acid. So very rare. Okay, if that happens here, that's not a good thing. Succinamide. So I give you a few examples. There's a lot of differences that happen. Then there is one small little Y that you see inside this antibody. That is a sugar molecule. So almost all antibodies have a particular glycan. That structure is called the glycan structure. They have a glycan in there. That glycan, and I will show you the structure of the glycan in the next slide just to give you an idea, is not a single type of molecule. It's a family of molecules. It's not a single molecule. Okay. And if you go to the right side, you will see there are two glycan molecules, right? In any antibody, there are two. This molecule can be different from this molecule, which means that if I have 10 of these and 10 of these varieties, 10 into 10 becomes 100. So I can technically have 100 different antibodies purely based on the sugar that is present. Does this have any function? It actually has functions that determine how Fc binds to the receptors, how it binds to macrophages, etc. Okay, so this is a very, very critical part of the antibody. Just to give you an idea, here are the structures. So this is a generic structure of an sugar that is present on an antibody. At the base is, so this wiggly line you see here, that is the protein structure. That's the protein to which it is attached. And here is your N-acetyl glucosamine. Then you have three mannoses like this. Then you have another N-acetyl glucosamine, two of these. You have galactose and you have sialic acid. Not every sugar is complete like this. So different sugars. Here is an example of the different types of sugars that are present in erythropoietin. Now I said that I will talk about both the proteins and vaccines, and this is the spike protein from CoV2. And every one of these round blue colored structures that you're seeing is a sugar molecule. Okay, very similar, but could be any of these different structures. Now you can imagine one company is making a spike protein to produce an antibody or a vaccine. Another company is producing the spike, but its spike is present on a virus particle like adenovirus. It is going to be differently glycosylated than the one that produces the pure protein. Now, which one is more antigenic? Which one will produce a better immune response? Very difficult to say. Therefore, you do the clinical trial, and what we are seeing every so often, that AstraZeneca's molecule looks good, Bharat Biotech's molecule looks good, Serum molecule looks good. Each of them, what they're saying is, we have tested our particular spike protein in whatever context it is there, and it is able to produce an immune response that prevents the virus. So each one does a different thing. So I've given you some background about protein structure, protein variability, etc. So again, but I haven't still stated what the problem is. The problem is that you need, if you are a biotech drug company, you need to make a biotech drug. Okay, that biotech drug has to, you have to take a potential drug, convert it into an actual drug. You take a product and you have to make it commercially viable. See, if I produce a drug which nobody can afford to buy, it's not very useful. Okay, This is something that you need to be very, very aware of. Okay. The third part is assure the properties of the drug do not vary from each batch. So there is consistency from batch to batch. One of the things about a drug company is we don't produce one batch or two batches a year. In Dr. Reddy's, we normally produce close to about 600 batches a year. My interaction with the regulatory bodies, which basically ensure that the product we sell is of the right quantity, is maybe once or twice a year. They come, they inspect us, they make sure that we do everything right. Why are they present once or twice a year? Why don't they sit every time? Because it's impossible, right? Which means what? The onus of making the drug properly lies with the company. It's not the regulatory group which makes it ensures that the drug is proper. The company that is producing it has to make the drug proper every batch and make it consistent. Okay. The other thing we need to do is we sell the drugs, we make it today, we sell the drug maybe tomorrow, maybe 6 months down the line, maybe 2 years down the line. Over the entire time, it has to be stable. So this is technically what the problem is. The problem is we as a biotech industry has to take a potential drug candidate, actually make it into a drug, make it commercially viable, which means that we should be able to sell it for a profit. Remember, we are doing this so that we can make money. At the same time, we can put some money back into research so that we can make drugs consistently over time. Okay, that's the expectation. Then we need to do this regularly, day in and day out, batch after batch, and this drug has to last for a couple of years at least, minimally. That is the problem. So what do industries do better? Industry does few things better. One is it knows how to break up the problem into small bits. In an academic setting, I'll just click through this so that you get a quick view of it. In academic setting, usually you go to a professor, they start the research, all the work is done in the same lab, and they publish some data that they have generated. In a biotech industry, we don't necessarily have one lab which does everything. We break up the problem. So let's say show you a very simple breaking up of the problem, right? What is a biotechnology derived drug from a very simplistic view? And I'm giving you a very simplistic view. It's a recombinant protein. Okay, that's the thing. Whenever we say it's recombinant, where is it produced? It has to be produced in a cell. Sometimes we produce it out of a cell also. Not everything is produced inside the cell. Okay, so at the end of it, is it ready to use? The answer is no. Why is it no? Because apart from producing your drug, the cell has produced a lot of other things. So then do we need to purify it? Answer is obviously yes. Is this purification predictable? Is it practicable, which means is it practically possible to do this? And is it commercially viable? The answer for all three are yes. Obviously, if it is no, then we wouldn't have a biotech industry today. And can we derive its quality? What I mean by saying can we derive its quality, it means that can we test it out and demonstrate that it is meeting the quality? Is it sensitive enough? Are the methods good enough? The answer depends. The reason I say the answer is depends is, and I don't say an absolute yes, is it all depends on the scientist who is developing the methods. And I can tell that with confidence because most of my life I have been heading the analytical development department. I develop methods to test products. Okay, so I know that if you develop the wrong method, you cannot find out what the real quality of the product is. So we have to be very, very careful. So there's a huge responsibility on the analytical team. So we normally divide our functions into what is called a cell line development, cell culture. I will explain the differences in the next few slides. Purification and analytical sciences. Okay, let's go to cell line development. The cell line is the ultimate raw material. What I mean by this is, see, anytime you produce a drug, whether it's a small molecule or a large molecule, you use raw materials. Okay, the raw materials then finally produce a drug of interest. In this particular case, for us, the drug is produced by the cell. Therefore, the cell becomes the most critical raw material. It is actually listed as a critical raw material. How do we do this? Every one of you who is in the biotech space knows about recombinant DNA technology, genetic engineering, molecular biology, all are the same, they're different names for the same thing. And out of maybe 1,500 or 2,000 students who I've interviewed over different years, 90% of them tell me that they want to do molecular biology. which is this one. Molecular biology is probably the most the smallest division in an industry, because once you produce the cell, that's it, you're done. So what does a molecular biologist do? They take the gene of interest. If it's an antibody, it has both the heavy and light chains are different genes. We take the DNA, put in the right promoters, the right stop signal, introduce it into the cell. It could be a bacterial cell, it could be a mammalian cell, and I'll go into details a little bit in a couple of slides. And then we wait for the transformation to happen. The cell is transformed, it produces our product of interest. Okay, so that is the basis of this. If you take a vaccine, is any different? Not really. I mean, there are some differences in vaccines and in protein. The difference is, instead of producing a protein, sometimes, like if I take the adenoviral vector, you produce an entire adenovirus. It's a defective virus, so you don't need to be worried that you're going to get an infection. At the end of the day, it's a recombinant cell. The cell is called HEC293 cell, and it's a kidney cell. You grow that up with a recombinant shuttle vector, just like you would do for a microbial system. Once you put it in, it produces an adenovirus. It's a defective virus, so it has no adenoviral nucleic acids inside it. But on the surface, it expresses your protein of interest. In our current recent experience, it is expressing the spike protein of COVID. So that is also a recombinant stuff. So vaccines are not really different. Even they have a cell line from which we produce it. Actually, vaccines sometimes have 2-3 cell lines, as opposed to, say, an antibody which has a single cell line by which we produce it. Okay. Now, understanding a cell line. Are all the cells identical? You must excuse me. I have one small problem with my computer. I will re-log in. It's gone blank. Give me 2 minutes and I will log in back.
Yeah, sure. Students, sorry for the delay. Within 2 minutes, Sir will rejoin. Sir facing some laptop issue. Please don't left from the section.
I hope you can see me again.
Yes, sir.
Yeah. Let me share my screen again. Apologies for this. Uh, I can't do much about it. Okay, so we are back on. So understanding a cell line. Are all cells identical? That is, are they all a clonal population? The answer is certainly not. If you transform a cell, when we do most of you have done transformation of E. coli as part of your genetic engineering or molecular biology lab experiment. You take a lot of plasmid DNA, you put it into many cells, some of them get transformed. Okay. When you try to grow this up, do you grow only one cell or do you grow everything? Normally you grow all of them, right? The problem is, in a plasmid based system, normally what you end up with is a plasmid which is inside the cell. It is expressing, but not every cell in that one has the same number of plasmids. And there may be that one cell has a genomic mutation whereas another cell does not have it. As soon as you have a situation like this, these two cells are different. Now imagine in a mammalian cell, you did a transformation, the DNA has gone into the chromosome. It has got integrated into the chromosome because most mammalian cells are integrated into the chromosome. You don't have a plasmid. Now you have no idea where it went into the chromosome. So if I get say 100 different clones, are they all the same? The answer is no. So can I take all of them and mix them together? The answer is no. I should always have separate. Now the question that you have to ask yourself is why do I know? Why do I care if it is one single clone or different clone? And how do I find out? And I'll come to the answer in a couple of slides. Next, I need to know, is the construct stable? Why do I care about stability? Because very important. Uh, if the cell is not stable, you can't produce the drug for many years. We need to produce the drug day in and day out, every batch, so that the market and the patients can get the drug. If the cell line is not stable, it's not possible. Therefore, stability is a very critical step. Then how do we measure stability? And what if we lack stability? What if we lack stability? You don't produce a drug. The regulatory bodies will never approve your drug. So we need to measure stability. I'll come back to that one also in a couple of slides. And we have to be free of adventitious agents. What I mean by that is, if you know, each of you when you work in the lab, you may have worn a lab coat or you may have worn a gloves. What you don't realize is that lab coats and gloves don't really stop you from infecting the culture. It's not the culture always infects you. You can infect the culture. Some of us have colds, some of us have cough, some of them have a skin infection. Some bacteria or some virus from our body can enter the culture. And if it enters the culture, what happens? The culture has very rich nutrient medium. It grows along with our product. And when you give it to the patient, they don't only not only get your drug, they also get an infection. So it's very dangerous. So our measure what the cell has and to make sure the cell does not have any viruses. We do clonality. In microbial systems, you know it's very simple. You do a streak plate, and when you finish streaking, you end up with something that looks like this. Uh, and when it looks like this, you can very easily pick up one of these colonies and say, I have a clone. Usually, you would do the streaking twice to ensure that you don't have two colonies right next to each other, but you didn't realize. Okay. In mammalian cells, the way we do this is we take a mixture of different clones and we dilute it out such that on a given plate you barely have 50% of the plates with even one colony. And normally we aim for half a plate being empty. In this case, one of these or few of these will be clones. We do this 2-3 times. We are assured that we have a single type of cell. That is called a clonal. That's therefore this arrow. It is done 2-3 times repeated, and you are assured that you have a clone. Now we come to measurement of stability. How do I measure stability? The number of copies of the gene. Do I have the equal number of correct number of copies every generation after generation? Why do I care about this? Because if I'm losing DNA, then today I produce X amount of protein, tomorrow I will have less than X. Is that a concern? It's concern you both from your industry because you want to be profitable, and it's also concerned because if I were to purify this protein, next time I will not have equal amount of protein. Maybe I'm going to purify some impurities. So we have to be careful. Next, where is it located on the chromosome? That is one way of showing that if I show I have 10 different locations and every time I have the same 10, it is very stable. I have consistent expression. If you do an expression study, some of you have run an SDS page after expressing, you can see the big band. That means that you have a gene of interest being expressed. Okay. Then finally, if I say integrity of plasmid, if it's extra chromosomal, if it's a plasmid, how do I do it? I make sure that I do a restriction digest, and if the restriction digest looks the same every time, I know it looks good. Okay. Finally, is the consistent impurity profile. Are all the other proteins that are present consistent? If other proteins are varying too much, then means your protein is also varying too much without realizing it. So these are all indications of how you measure the stability of a clone. And these are the adventitious agents. I just given you a quick snapshot of how many different types of viruses we test for, making sure none of them are there. Some of them are familiar for you. You have polio virus, you have hepatitis A, you have the HIV virus, then you have murine leukemia virus. A lot of these we test. And how do we test for this? We do either in vitro and in vivo assays. We do something called the reverse transcriptase assays. We do electron microscopy, we do PCR, different tests. Different tests are done in industry. So this is all done by the cell line development group. Okay, they develop a cell which does not have a virus, has got the gene of interest, which is very stable and produces a product for us. That is the cell line development group, the molecular biology group. The second group is the cell culture development. What do? They take the sole purpose of cell culture is to produce the maximum amount of product with a major portion of the product being of the right quality. Okay, that is the job of the cell culture. We have scales. If you take, if some of you may have gone to the lab to see a fermenter, normally you would have seen a 5-liter fermenter or 10-liter fermenter. Actually, fermenters come in the smallest size at 10 ml, and in India, I think the largest one is about 1 lakh liters. So you're talking of orders of magnitude difference. Okay, that's the kind of fermentation capabilities that exist in the world. Most antibodies are produced in the 2000 to 10,000, 20,000 range. Antibodies normally are not produced above 20, 25,000. Very rarely you go above that. Insulin is produced very regularly, like in Biocon, they have a 30,000 and a 50,000 liter reactors for insulin production. Just to give you an idea, here is a bioreactor. Some of you may be familiar with this one. This is 2-liter bioreactor. The same thing you can do in a 15 ml bioreactor. It has all the same settings. Here is, so this one, this is called the Amber. For those of you who are interested, it sits inside this. This single machine can hold 24, 48 or 72 Amber machines. These Amber reactors. So 72 different fermentations can be done. What does AMBR stand for? AMBR stand for an advanced microbioreactor, which basically means it's a small reactor which has all the features of a big reactor. And you can develop processes by this. This is how the cell culture team works. It takes, because the media is very expensive, instead of growing 2 liters, if I have to run 72 reactors of this much, I need 144 liters. I can't spend that much. And if I do say 10 or 15 ml, in a liter I can do all of these reactors. So you can imagine the cost is almost 100 to 200 fold less. That's the advantage of running these reactors. And they can be instantly transferred to 2 liters and they give the same results. Okay. What are the challenges? One is media preparation and sterilization at scale. In a small scale in a lab, most of you would have done a small autoclave in a shake flask, maybe a 2-liter shake flask. When you are growing things at say 20,000 liters, what are you going to do? How are you going to sterilize the media? How are you going to remove the heat after say autoclaving? Are you going to be taking the reactor to the autoclave, or is the reactor also an autoclave? Right? These are things that you will learn in industry. There are big reactors which themselves are autoclaves in many senses. They sterilize it with steam at the location. Okay, so this is a very critical part of an industry. The recombinant proteins usually are derived as a plasmid. So if you have an expression system, if you have a very famous one which you guys are all probably familiar with is the gal operon. You induce it with the lac operon, not the gal. You induce the lac operon with IPTG. When you induce it with IPTG, you add a small amount of IPTG. In a say 10,000 or 20,000 liter reactor, how much IPTG are you going to add? How are you going to add it? In a small tube? How long it will take to mix? Will all the IPTG be consumed by the cells at the top? These are all questions that have to be asked. And antibiotics in a manufacturing system are not acceptable. You remember when you grow cultures of E. coli, you always add antibiotic like penicillin or kanamycin to maintain the plasmid. In this system, you can't add it. You're not allowed to add. Why are you not allowed to add? Because sometimes when you finish purification, you may be giving the patient this antibiotic along with your drug. And people are allergic to antibiotics like penicillin. And if your patient gets antibiotic allergy, they will probably die from the medicine you're giving than from the medicine helping them out, right? So antibiotics cannot be used in a manufacturing process, which means what? The plasmid has to be stable without antibiotic. And that's not easy when you're growing up things in 10, 20,000 liter reactors. Okay? Then induction of gene synthesis can be triggered when cells reach high enough cell density. See, normally what do? We take a culture, we grow it up, say OD1 or OD2, and then we add the IPTG and we shake it up and it continues to grow for a little while. Say your boss will tell you, let it grow for 2-3 hours. You grow it up for 2-3 hours, you purify the protein. In industry, we want maximum number of cells. One OD of E. coli equals about 8 times 10 to the 8 cells per ml. We do in industry, when we induce it, the density of the cell is about 50 OD to 100 OD. Sometimes you can even go to 200 OD units, which means that the cell culture is almost like a paste. It almost looks like cake batter. So to be able to induce at this stage high enough cell density is not a trivial problem. All of these are engineering problems. How do you design a reactor? You have to control the oxygen, which is called the KLA, the oxygen exchange rate, the pH, the temperature. How do you keep the temperature going up, going down? Everything has to be controlled. And finally, you need to extract out the protein after you grow up the cells. You extract out the protein. Is the protein expressed inside the cell or is it expressed outside the cell? In most microbial systems, we express it inside the cell. In mammalian systems, you express it outside. So all of these problems are common for microbial cultures, okay? Both intracellular and extracellular. Now, if I come to a mammalian culture, the challenges are very similar but slightly different. Cells are very, very delicate, okay? If cells are very delicate, how do you grow them to high cell densities? Actually, currently there is a new technology, and I'll speak about that in the next slide. There are ways in which you can get high cell densities, but the problem with delicate cells is you can't grow them very quickly. You can't, agitate them very fast. Therefore, a microbial reactor and a mammalian reactor are very different. Media sterilization. In microbial media, normally we have a lot of different salts, we have glucose, all of them can be autoclaved. Mammalian media, you can't autoclave it. If you can't autoclave it, how are you going to sterilize it? You have to filter it. That is a big concern. Okay, filtering 10,000 liters. What is the scale of filter do you need? How do you make sure that even if you get the smallest contamination, remember something, a mammalian cell doubles every 24 hours. Whereas bacteria can double theoretically every 20 minutes. Which is before the mammalian cell culture even grows, the bacteria would have used up all the media, it would have been, your culture is destroyed. Therefore, keeping it sterile is very critical. Then mammalian cells usually the culture lasts for 10 to 15 days. That's the culture duration, okay? Just a second. Whereas in microbial systems run for only about a day. So when you're doing 10 to 15-day culture, we need to first make sure that we have enough food for the culture, for the back cells for 10 to 15 days. It means you may need to add food every so often. And the food is that you add could be amino acids, it could be sugars, it could be vitamins, anything. You make sure it's sterile for 15 days, that no bacteria enter it. And you need to make sure that the cells are viable for those 10-15 days. What are the things you need to control in mammalian cultures? You need to control oxygen rate, you need to control CO2, you need to control ammonia, you need to control osmotic strength, temperature, pH. Now some of you may ask, why do I need to control osmotic strength? If you remember plasmolysis, when you add salt to meat, the meat lasts longer because you removed all the water from the cells. Same thing will happen in a cell culture. If you have too much salts in your cell culture, instead of the cell growing, it dehydrates. In which case, all the cells will drop dead. You have no product. So controlling osmotic strength is very critical. And this is why when you give an injection to a patient or when they give a drug, they give it as an IV. For those of you who have seen an IV injection, they give it in saline. What is the reason they give it in saline? Why are they mixing it in, say, dextrose or saline? Because that has the same osmotic strength as your blood. Okay, remember that. Okay. Then when you talk about cell culture, you have to think about how proteins express cells. Okay, in microbial system, induction is carried out at the last generation. Cell viability is not relevant. You grow up the cells, last doubling of the cells, you add the inducer, it produces the protein. Whether the cell is alive or dead, you're done. Whereas in a mammalian cell, your culture has to be 10 to 15 days, which means it better not be dead on the day one. It has to grow for continuously, and it keeps producing product. So how do you measure protein productivity? In mammalian cells, you measure productivity by two terms. One is called QP, which is the specific productivity. Simple terms, how much protein does one cell produce in one day? That is QP. And you take it and you multiply it with the number of cells that have been present throughout the culture. This is called the integral of the viable cell count. What is the viable cell count? You know the cells have a viability. Here is an example. On day one, just assume this to be I have one cell. Okay. At the end of day one, the cell now becomes two cells, which means that on day one I had one cell, on day two I have two cells. Total number of cells, cell days, 3 days. 3 cell days are there. If each of the two doubles now, you will get 4 cells. So 4 plus 2 plus 1, which is 7 cell days. So that is what IVCC is. In that case, it will be called 7. That is, if you have millions of cells, how many million cells were there over the entire period? That becomes the IVCC. And each cell is producing a specific amount of protein, that is QP. You multiply these two and you end up with the total protein produced by a cell culture media. Okay. How do I increase? Remember, I told you this that in this slide I said cells are very delicate. How do I make them grow more and produce more protein? There is one technique called the alternating tangential flow filtration, which basically this is a reactor. Okay, just assume this to be a reactor. Does not matter what size it is. It could be 10 ml, it could be 10,000 liters. You're growing up the culture. The reason you are taking 10 to 15 days to grow it is because it grows slowly, 24-hour doubling, but it also produces toxins. Okay, as it is produced, growing up, it is throwing out all the garbage that it has used up. This will stop the culture from growing very much. So what you do is you take some part, one small portion, let's say 5% of the media, every day, remove it and throw it out and add fresh 5%. When you do this on a continuous basis, your media is becoming fresh every day, and fresh media is being put in, old media is being removed. Here is this is actually a very sophisticated filter. What it does is it takes the media from inside the culture, brings the media along with the cells. The media is filtered out, the cells are returned. Okay, so only the media is being thrown out, the cells are being returned. In this case, you are now ending up with more and more cells, and this can actually increase the cell density significantly, and you get a huge increase in the amount of protein. Normally, we produce about say 1 to 5 grams of protein per liter. By this one, you can go up to 10, 20 grams per liter. Okay, that's a very significant increase. Okay, so what does cell culture finally define? The cell culture is able to produce a product that is properly folded. It has got the right charge, it has the right size or fragments if it is. All of the glycosylation, glycation, acetylation, deamination all are under control, and it produces enough protein to make it commercially viable. Okay, this is what the cell culture group does. So I finished with the cell line development, which produces a cell. The cell culture group does this. It produces a protein of enough yield, which has all these product properties that you are looking for. Okay. The third group is the purification group. Whether it's proteins or vaccines, they're very, very similar. Okay? Purification is a subtractive process. What I mean by that is you can't add new characteristics during purification. Whatever cell culture gives you, you remove all the impurities. Therefore, it's subtractive. What you are left with is good product. Okay, very well developed in specific areas. Certain areas of purification are very easy, very straightforward. What are the levers we use for separation? We use charge, we use hydrophobicity, we use affinity, we use size, we have differential solubility. Okay, you guys are all be familiar with the terms chromatography and filtration. Whether it is chromatography or filtration, pretty much these are all the properties that are used. Incidentally, even for filtration, you use similar properties. Some of you may think, hey, filtration means only size. Not necessary. You have filters which have charge on them. They will allow certain things to pass through and bind certain things. They may be hydrophobic filters. Okay, so you can choose the filter membrane also to define how you are going to purify. This is done as part of the purification scheme. Okay. What are the critical areas that purification stream does? Host cell proteins, host cell DNA, any other host cell impurity, they have to be removed. That is very, very. Some proteins are very finicky. Okay, there are some proteins which do not fold properly, that tend to denature easily. To purify these proteins is very, very, very difficult. Probably one of the most difficult things to do is to purify such kind of proteins. So the process, the purification development team struggles with proteins like this, and it's a big challenge for future scientists. Then a lot of work, see, all the cell culture work is done inside a reactor, whereas the purification is not done inside a reactor. It is done usually in the there are filtration skids, you have chromatography columns, etc. But there is some amount of open air operations. So you can easily end up with a virus coming from a human being. So you have to be very careful at scale how this is done. Make sure that no non-host cell protein. See, you are always testing for host cell proteins. You're not testing for proteins which you don't know exist. So if a bacteria falls in it, you don't know. So you have to be very careful that no bacteria falls into it. Okay. And what's the normal process? In an say if I take an antibody, very standard. You first have one chromatography, then you do a inactivation of any viruses. By mistake, if a virus came, let me inactivate it. That's a precautionary step. Then you do a filtration, and is called UFDF for ultrafiltration diafiltration. Basically, because the volume is very big, you concentrate it down. Then you put to one column, a second column, two different exchanges, ion exchanges. One is anion, one is cation. You got a fairly pure protein. At this stage, it is 99.9% or 99% pure. You do one viral filtration. You then do one more concentration, depending on how concentrated you want your drug. You're done. So this is a very standard purification scheme. Now here is, I just showed you the standard purification scheme, just a different image. Vaccine looks more complicated, but not that different. See, you have raw material preparation, amplification, all of this is upstream. Once you have done that, you filter it, remove the bioburden, you remove nucleic acid digestion, which is host cell DNA. Then you do a concentration, which is the same as filtration. Then you remove any more bioburden that may have come during this process. Then you do chromatography 1, chromatography 2, you do filtration. If you have thing, you're done. So very similar. So this purification scheme is very similar to this. So whether it's a virus for a vaccine or a protein, purifications are very, very similar. Okay. Then we come to the last of the four big chunks of product development. It's the analytical sciences. Okay. What do the analytical sciences do? One very, very, very important point for you guys. So those of you who are in the process development, it is important. Who like, and the reason I'm separating out this is some people will be interested in molecular biology. Some of you will be interested in cell culture development. Some of you will be interested in purification. And some of you will be interested in analytical sciences. Incidentally, I have been an analytical scientist most of my life. I was head of analytics in Dr.Reddy's for a long time. So this is my area. And I will spend the least amount of time probably on this one because I don't want to scare you with too much information on this one. Okay. Just want to let you know that testing is not a substitute for a poor process, which means when a regulatory body comes, they are always interested in how you produce the drug. They're interested in how you test it, but that is less important than how you produce the drug. The reason that is less important is because no matter how good your testing is, your product quality is only defined by how you produce it. So it is very important to produce the right amount of drug, and testing is not a substitute. Okay, so it has to be a good process. You can never test for something unless you suspect that it exists. If there is a virus that I did not know, I am not testing for it, so I will not find out that there is a virus. So you have to know and be aware of this. This is very, very important for you guys to understand that awareness from an analytical scientist is a very critical fact. What do you expect? What do you think is there in this particular thing? What impurity could be there? And this is very true for all the tests that you're developing. You have to begin to start based on the science that you learned, based on the biochemistry you learned and the protein chemistry you learned. You have to ask the question, what impurity can be present in this stage of my process, and develop a method to test that particular impurity. The other thing you need to understand is risk. Is it an risk is understanding risk is an essential skill for pretty much all scientists, specifically for analysis. Because see what happens is my process development team produces a drug. I am the person from an analytical person who tests the drug and says that this drug is good to be given to the patient. So at the end of the day, my analysis has to be of a quality by which I can assure the regulatory group that look guys, this is good enough to go into a human patient. If you do not have that ability to understand what is the risk associated with this, what kind of impurities are there, you will always be nervous to take that decision. Can I inject a human being? That will not work. So you have to understand protein chemistry very well. You need to understand impurities very well, etc. Okay. Then uncertainty of measurement is compensated either statistically or orthogonally. What I mean by this one is not every measurement is going to be very accurate. Some measurements are inaccurate. Okay, that is the nature of measurement. Any measurement, give the most accurate measurement has a certain amount of residual error that is given. How much is that error? Is it worth worrying about that error? Is there an alternate method by which I can also test and reduce that error? These are decisions you will take, and that's a decision that will help you. Statistics helps you take this decision. What I mean by statistics help you take the decision is if I have one method which is greater than 99.9% ability to predict something and another method which has got another 99.9, it is not additive, it is multiplicative. Okay, that both the methods would have missed an impurity becomes multiplicative. Therefore, the purity can become 99.9999, which is the kind of assurance you want. Okay, why I wrote meter dosing. Imagine you're giving a person an insulin injection. Okay, now insulin injection the doctor recommends 12 units, 14 units, 16 units. If your measurement is not right, somebody who is supposed to get 12 units gets 16 units or gets 10 units. That's not a good thing. So the ability to measure a concentration and the ability to tell what is the error of my measurement is very, very, very important. So this is something I want you guys to keep in mind. Okay, and analyticals, actually the analytical sciences guides the entire process. Whether we are doing testing of each interim step, you remember in the upstream and in the purification I told you there are multiple steps that are involved. In each step you can take a small sample and measure. You can measure different properties. That's called in-process testing. When we do the final drug substance or the API testing, that's called lot release assays. And over the shelf life of the product, we test stability. So we are, as an analytical science group, remember something, you guys will be testing the product across different time frames, and you will be then assuring that, hey, the process and the product is of right quality. Okay. How do I do that? Analytically, you always break down a product into smaller and smaller chunks. The smallest chunk is called the primary structure, which is the sequence. You can measure the sequence, but you do that only once. When the cell line is being developed, you measure this actual sequence of amino acids. You derive the sequence using different methodologies. Once that is done, you don't need to do this repeatedly. But on a routine basis, we measure the secondary structure, we measure the tertiary structure, and for those proteins which have a quaternary structure, we measure the quaternary structure. So something like an antibody has a quaternary structure. GCSF does not have a quaternary structure. Okay. Then next, this is a slide I took from the FDA website. Actually, they're not website; they have a presentation. This blue symbol here is the FDA symbol, and it says the different types of analytical tools to be used to evaluate a protein. Almost all of these are done by my lab. Not every one of them, I will guarantee you. We don't do this HDX. A very complicated method, but has very little value in my opinion. Okay, this is my personal opinion. We don't do NMR very much. We don't have an NMR machine, and we don't even give it to other companies or industry academics to do it because NMR results, like I showed you the GCSF structure, it gives you important information, but not on a batch to batch basis. It's not needed. Okay. So what are the kind of things we do? I'll just quickly walk you through. I'm looking at the time. I have another 30 minutes of talk. I won't take all 30 minutes. I'll give you more time for questions. Okay. So we do the amino acid sequence identification, and if there's any modification, you can do that by mass spectroscopy. MS, anywhere I've written MS, it's mass spectroscopy. You do that by peptide mapping. For those of you who are not familiar with these terms, let me just quickly walk you. I think most of you will not be familiar with a lot of these terms. So I'll give you a brief couple of them so that you feel a little better. Peptide mapping is a technique where you take a doesn't matter which protein, if it has single subunit or multiple subunits, you can do it for both. Uh, then you digest it with certain proteases. Example being, say, trypsin. Okay, if you digest it with trypsin, what happens is it breaks it up into smaller chunks. Then each of this total digest is then the total digest is put on an HPLC, normally a reversed phase column. When you put it on a reversed phase column, you will end up with peptides that are separated out from each other. These peptides will form a very characteristic pattern for a protein. No two proteins will give you the same peptide. Therefore, it is called sometimes peptide map fingerprint, the fingerprint of a protein. Actually, one way of identifying proteins is by just taking a random sample and digesting it with a protease and finding out what peptides are there, and then back checking to see whether I know this protein. Okay, so peptide mapping is a very common method of identifying the integrity of a protein. There is chromatographic separation you can do. Amino acids directly, if you take a protein and boil it in acid, weak acids, you can hydrolyze each of the amino acids separately. Then you can separate out those amino acids in a chromatographic column. Therefore, you can identify what are the amino acids present in a protein. Folding. How do I identify folding? You can take, say, disulfide bonding. You can do disulfide bonding. You can check that by a particular chemical which actually breaks the disulfide and then adds a fluorescent tag to that. Okay, so there are ways you can measure how many disulfides are present. You can measure the stability of a protein by something called calorimetry, which basically tells you that if I add a certain amount of energy to the protein, how much does it warm up? And if it heats up in a specific manner, what am I doing when I'm heating up a sample? Bonds are absorbing the energy, then bonds are breaking. So very characteristic of the structure of a protein is how they're bonded together. You remember your hydrogen bonds, your covalent bonds, your ionic bonds, all of these bonds break at different amounts of energy. Based on the energy, you can identify the protein structure is stable or not. Then from folding, I will go to, say, subunit. You guys are very familiar with chromatography. Size exclusion chromatography or gel filtration or gel permeation, whatever names you use, it's all the same thing. There you can tell whether a protein is a single subunit or multi-subunit. Pretty easy stuff. Then you get size, aggregates, and charge. If you take this is you can, other than chromatography, you can do, say, scattering, light scattering. You know that light wavelength is small enough that it can scatter small proteins, and if a protein is an aggregate, it will scatter the light differently than if it is a monomer. Based on the scattering, you can actually measure the scattering, and you can figure out, back calculate the size of the protein, whether it is aggregated, whether it's a monomer. All of these can be calculated out. Okay, you can also use something called analytical ultracentrifugation, which is basically a technique where you can spin the protein at very, very high speeds on a density gradient. The density gradient can be made with, say, sucrose. If you have very high concentrations of sucrose and centrifuge it very much, the bottom of the tube actually has a higher density than the top of the tube. And then if you layer a protein on top and then you do the centrifugation, if the protein is a monomer, it will go down to say some extent, whereas an aggregate may not enter the solution very much. So you have certain amount of aggregate, then you can measure that. So each of these methods that is listed here gives you a unique property for the protein. Okay, you cannot do only one technique and be satisfied. You have to do most of these techniques, not necessarily all of them. And as a scientist, you have to decide which ones am I going to do for which protein or for which vaccine. Okay, I'll give you one example of this one. There is a big push on trying to find out the size of a protein. and whether the protein has aggregates. So now if you take a look at it, normally proteins are in nanomolar range, okay? That's the normal size of a protein. If it aggregates, it forms about 10 nanometers of size. If it becomes bigger and bigger than this, it becomes subvisible, means you can't see it, it's in the micrometer range, but it's now big enough that actually hundreds of proteins are aggregating together. And then if it becomes very big, which is almost a precipitate, you see a visible particle. That is a precipitate. This you can actually do by even by a person who has got very good eyesight, not somebody like me at my age, but somebody who's like say in the 20s, 30s, good eyesight. You can measure, you can see this. But for this, you need instruments, and these are all the different types of instruments that can be used. Okay, that's the reason why this, and why is there so much importance with size. It turns out that protein aggregates have a tendency to cause immunogenicity, a very dangerous reaction. You may say, hey, why is immunogenicity dangerous? Imagine there is a patient who is getting erythropoietin injection. Okay, erythropoietin is necessary for blood cell creation, red blood cell creation. It actually stimulates red blood cell synthesis. If I have an aggregate of erythropoietin, suddenly my body recognizes that as a foreign particle. I might end up with an immune reaction against erythropoietin. In which case, what happens? The next time you try to give a person an injection of erythropoietin, or your own body produces erythropoietin, antibodies will kill the erythropoietin. Suddenly, the person has no red blood cells. The person drops dead. You may be thinking this is theoretical, but this is actually what has happened once. Actually, twice it has happened. Once in the case of erythropoietin and once in the case of darbepoetin that I know of. It's called PRCA, for pure red cell aplasia, where patients' blood actually became colorless, white. They died because they lost entire blood from their body. Because erythropoietin was antibodies were produced, and they could not produce any more red blood cells. So getting these techniques down is very important. Okay, so now I will just summarize the industrial part, takeaways from industry. So what does industry do better than an academic? You remember I said started off saying what does industry do better than academics? We are very good at specializations. Okay, we believe that the best way to address a problem is only by breaking it down into specializations. Now, does academics not have specializations? Absolutely not. They do have. They have great specializations. There's biochemistry, there is molecular biology, there is, there are especially institutes which do just structural biology, the institute which do biophysics. So, entire academic institutions and academic departments exist which are specializations. But within that specialization, if you go to any professor, they do the entire gamut of work that happens. So they may grow up the cells, they may purify the protein in small scale, but their focus is, let's say, biochemistry. Their focus is crystallography. And I can say that because I did my postdoc on crystallography. I grew up the cells, I purified the protein, then I did the crystallography. That is the focus of the lab. So there is a specialization that there in industry where they won't allow you to do it, so you become really good at what you're doing. Then when we break it down, it leads to excellence. Industry has to have excellence, and I'm not saying academics don't have to have excellence. Obviously, academics have to have excellence. Unluckily, in the Indian scene right now, we still are struggling a little bit on this aspect. Industry has a lot more excellence associated with it, and you may ask, why is that so? There is money to be made. Because there is money to be made, we force excellence. If you ever have a chance to visit an industry, you will see that the kind of equipment we have is the best in the class. We have the same equipment that most companies around the world have. India has Indian industry. You walk into the lab and you will feel you're outside India. The quality of instruments is, they're all imported of the highest quality, because we expect to sell our drugs all over the world, so we have to be excellent in what we do. In industry, science and engineers work together. Scientists and engineers, not science, I'm sorry. Scientists and engineers work together, okay? Because scientists are good at doing certain things at a certain scale. Engineers are very good at converting that into large scale, manufacturing scale, production scale. There, it's not a scientist's job, that's the engineer's job. So let them do their job, let us do our job, and we work together and you become successful. So now that I've given you a brief on the biotech industry, and I just alluded a little bit on the vaccine, because when I was looking at making this presentation for vaccines, I realized that giving you only a story on the vaccines as being something special would not have done it. The same thing that happened here in the biotech space, whether it's a protein or a vaccine, is the same. So tomorrow, whether you join a vaccine producing company or a pharmaceutical biotech drug producing company, the skill sets are almost the same. If I know somebody, I've hired people from vaccine industries, I've had people from my company go join the vaccine industry. We fit in easily, no difference. So from the next generation of scientists, that is you guys, okay, what are the kind of questions you guys need to ask? One is technical. Okay, I have three challenges for you. Critical challenge in biotechnology. One is technical. How do you improve efficiencies in process? The current cell culture processes produce drugs. If you take an average dose for an antibody, it's about 250 milligrams, which translates to about $95 per vial, which translates to about 21,375 per dose. Okay, 21,375 per dose. If I say that 250 milligrams, actually this is $95 per 100 milligrams. So for 250 milligrams, it would be about 21,000 rupees per dose. And say some of the drugs that we sell in biotech, patient needs say 10 doses. You're talking of 2 lakhs. Can everybody afford 2 lakhs? The answer is no. Do we have to do something about it? Yes, we need to make our processes better. So the challenge for you guys in the next generation is, how do you make the processes of cell culture and purification way more efficient so that the price can be dropped by almost tenfold? That's a huge challenge and it is there. Okay, challenge number two. This is an Indian challenge for you guys. This is an ethical challenge, okay? Should we edit genes or can we control the use of CRISPR? I know a lot of you would have heard of CRISPR. I did not want to talk today about CRISPR at all, because everybody wants to talk about the latest technology, forgetting that your foundations are more important than the latest technology. You will become good at new technology only if your foundations are good. But why do I bring this up? This is an this is I borrowed this from a science article in the magazine Science, which talks about the number of publications each year from 2012 to 2018 in the field of CRISPR. Okay, and you will notice that India has produced about 50 papers. China and US have produced greater than 800 papers. Kind of sad, but that's the truth. Now you take on the right side, this one is the number of patents supplied by each country. Okay, United States, China have about 800 patents each. These are patents, which means that they have converted it into an application. Is India's name even in this list? The answer is no. That reflects the fact that in Indian academics, industry is not doing anything on CRISPR because we don't know how to make money yet. But Indian academics have still got to figure out how to take these 50 papers and convert it into any kind of patent or money, which means that our ability to think of a problem, solve it, and make it into a product still lacks. Remember something I started off saying, that one of the things that Indian industry or industry does different from academics is to take a product from a theory and convert it into a product, make it commercially viable. This shows that we still lack this significantly in India. Okay, and this is a matter of concern that your generation of students has to solve. And while solving is, you have to have this ethical question. CRISPR can be used to do a lot of damage. Those of you who are not familiar with this technology, I urge you to read a lot more on this technology. You can actually design human beings with this technology. Is it easy? The answer is yes and no. The technique is easy, but is it easy to design human beings? The answer is no. But is it impossible? Again, the answer is no. It is certainly possible. And I would not be surprised if China is taking a lot of risks in this one. Should India be taking those risks? I would never recommend that. But we need to be aware of this. We need to be able to use this technique, to understand the technique, to do benefit to our people. Okay. This is something that your generation has to do. Finally, the critical challenge is the social challenge, food and fuel security. I spoke about all the biotech of drug industry. I didn't speak about any other biotech aspect. One of the biggest contributions, and incidentally, that is my postdoctoral work, etc., is on this aspect. I haven't done this in 20 years now, but genetically modified crops. Should we use it? Should we not use it? Is it worth doing? People are scared of GMO. You are also remember that global warming is real. If global warming is real, that's because we are using up all the fuels. So shouldn't we move to biofuels because they are recyclable? The question is how much. And here is an example. If you take the fuel requirements of the existing world today and say, I'm going to make the entire fuel come from corn, you would need about 172 liters per hectare, and the amount of land you would need to produce would be 1540 million hectares, which is 8 times the current land present on the entire planet, which means that you can't do this, basically, right? You do this through all the systems, and the only systems that allows you to do this is algae and microalgae with low and high yield. Their liters per hectare is in the range of 58 to 130,000 liters per hectare. The total amount of hectares of land needed will be only 2 to 4 million hectares, which is about 1 to 2% of the agricultural land of the world today, which is easily doable. So, will you go down this path? That's the social challenge you guys need to think about. Okay, so that is it. And now the long-term view of the field. Where do I think you guys need to focus? This is the last couple of slides. It's almost time. I think I'm on time. Yeah, it's 4:20. So a long-term view of the field. Here it is. These are things that scientists like you should be focusing on. Data analytics, understanding tools to evaluate large pools of data, a shift to predictive science from descriptive science. As you go in industry, you will realize that the amount of data available is humongous. The ability to understand this data and then use it to predict things will be important. It requires a knowledge on computer science, it requires a little bit of knowledge on statistics, and if you are not aware of computer science or computer languages, programming and science and a little bit of statistics, now is a good time to learn because data analytics is going to be one of the big things. Remember, India pretty much lost the race in say Google search engines. You know how much information goes into Google, Facebook, WhatsApp, doesn't matter which platform you use. Every one of these platforms is using your data and predicting things. To say that I'm a biologist and I don't understand it is not an excuse. The data that you will have is lab data, and to be able to analyze it is important. So one big area of requirement for industry for the foreseeable future is the person who can do data analytics. Somebody who can think automation. Okay, as international borders become easier and easier to travel, incentives for collaboration, international collaborations increase. The Modi government has been bringing in a lot of direct foreign direct investments, so the money is huge. But international means also that they expect high throughput, high sensitivity, quick turnaround time. All of this will be only possible if you can understand automation, you can work with automation. Okay. Then specialization and expertise. These are not new terms, but what you also need to focus on is international regulatory requirements and patentability. Is my technology acceptable by regulatory groups, and can I patent my product or patent my technology so that we can make money out of it? That should be your focus. If it is not patentable or if it is not worthy of getting a patent, which means you're either copying or you're doing something useless. Don't waste your time. And then again, as I just said, digital awareness is mandatory. The learning of computer languages, the learning of mathematics, the learning of these things becomes almost necessary. You cannot hide behind the tag of a biologist anymore. And last thing that I want to state is leave your Indian habits at the doorstep. What I mean by Indian habits, don't leave your Indian roots. Okay, never leave your Indian roots, but your Indian habits of saying chalta hai, mediocrity is okay, kal karenge doesn't work in India, in industry basically. You have to be on the job as quickly as possible because a day's delay means somebody else has won the race. In a patent situation, if your patent application comes even 2 hours late, you lose the patent. The person who sent it 2 hours ahead of you wins the patent. So be a very, very, very aware of this one, that these habits that we have gotten so used to of saying "Arey kal karenge, chalta hai, doesn't matter," you know, leave that. Only then will you succeed in the industry, whether it's biotech industry or anything. So I think I'll stop at this stage, and I'll be more than happy to take any questions from the audience. Is anybody there?
Yes, sir. Students, anyone having any queries, please put in your chat box. Please respond. Sir, one more question in YouTube. Can you tell me how to learn skill and programming language?
Uh, so a lot of the languages that you want to learn, programming languages, are actually available online. There are different ways of doing it. You can either be a self-taught one, or you can attend. There are courses that are offered by places called Coursera, Khan Academy. They teach you things. Khan Academy is free. Coursera also, some of the courses are free. If you were to take something like let's take an example of a thing called MATLAB. MATLAB, it's available on the internet for free. You have courses that you can walk through, learn on your own. So there are a lot of courses that you can learn online on your own. You don't need to join a college. You don't need to be enrolled in an institute. The more time you spend on the internet learning languages, the more profitability comes with it.
Okay,
Anything
sir.
else?
Yeah, one more question, sir.
No?
Sir, what is the main difference between Sputnik Q and convex?
I couldn't get the question. Difference between?
1 minute, sir. Sir, please click on chat box.
Yeah. Okay. Sputnik V and Covaxin. Okay. So Sputnik V and Covaxin. So I will talk about Sputnik V because Covaxin's details are not necessarily known. Sputnik V has two adenoviruses. Okay. There is an adenovirus 5 and an adenovirus 26. On the surface of the adenovirus, there is an expression of the spike protein. So this is a defective adenovirus which has the spike protein on top of it. What is done is, the reason there are two different adenoviruses is each patient, or each person, not a patient because this is a vaccine, each healthy person is given first one of the adenoviruses, which is adenovirus 26 if I'm not mistaken. What will you generate? You generate antibodies against the spike protein and the surface proteins of adenovirus. After 21 days, you get the second injection wherein the spike protein is expressed on adenovirus 5. There you will end up with antibodies against spike protein. Therefore, it is a booster dose, and adenovirus 5, which is new. So you won't have very big immune response against the adenovirus. So the reason you have two different adenoviruses is because the only common protein between those two will be the spike protein against CoV2, which will then give you a very significant immune response, which is your vaccination.
Yeah, thank you, sir, for clarification. Students, anyone having any query, please put in your chat box. Sir will explain you. Please respond, students. Students, please respond. If you are having any query, please put in a chat box. Students, please respond. Anyone having any queries, please put in a chat box. Otherwise, put no queries, no doubts.
That means that either I explained everything so well or you didn't understand anything. One of the two has to be true.
Please respond, students. YouTube, they are telling thank you, sir.
Okay. Thank you very much.
Thank you so much, sir,
Yeah,
for a valuable information from your side.
not a problem.
Thank you so much, sir.
I will log off now. Thank you.
Yeah, okay. Thank you, sir. Bye bye. Thank you, madam. Lakshmi, madam. Students, I have given day 5, day 6, and day 7. Last week feedback form also. Please check it and give the feedback forms without fake. Based on that only, we'll provide the certificates. Thank you. The section is closed.